In this video, I'll talk about my original research, Dissolution Without Disappearing, Multi-Component Gas Exchange for CO2 Bubbles in the Microfluidic Channel. The work is published in Lab on Chip in May 2014. The main idea of the research came from the original observation in the microfluidic channel. We used a flow-focusing geometry to generate CO2 bubbles in a stream of surfactant solutions and observed that the bubbles do not fully disappear despite the fast dissolution rate of CO2. Using both experiments and a theoretical model, we were able to explain the behavior of the CO2 bubbles, attributing it to the multi-component gas exchange between the bubbles and the surrounding liquid. Now let's look into the main story. In the main experiments, a serpentine microfluidic channel with a width of 150 micrometers and a height of 38 micrometers is used. By adjusting the gas pressure and liquid flow rate, we obtain a steady flow of CO2 bubbles that move downstream along with the surrounding surfactant solutions. We varied the concentration of sodium dodecyl sulfate, SDS, from 0.5 to 20 millimolar. For all experiments, the initial bubble radius was about 15 micrometers. Since the bubbles flow along with the liquid phase, tracking a single bubble allows us to observe stationary bubble dissolution within a box of liquid that is moving downstream at the same flow velocity. To analyze the influence of surfactant concentration, we plotted the normalized bubble radii versus time for different SDS concentrations. First of all, under all tested SDS concentrations, we observed that the bubble size decreases rapidly for approximately 30 milliseconds after generation, and then reaches a steady state value. Also, we observed that there is clear concentration dependence below the critical micelle concentration, CMC, which is 8 millimolar for SDS. Above the CMC, there was no concentration dependence on the dissolution behavior of bubbles. Finally, we confirmed with longer microfluidic channels that the equilibrium behavior of the CO2 bubbles lasts long. Considering the solubility and the diffusivity of CO2 in SDS solutions, we know that the equilibrium behavior is not caused by the saturation or local saturation near the bubble interface. Therefore, to better analyze the influence of various parameters, we set up a theoretical model that considers multi-component gas exchange. In the model, we consider stationary gas bubble dissolution in an infinite liquid phase. The preliminary calculation using a single component, which is CO2 here, cannot explain the steady-state radii of the CO2 bubbles as you can see in this graph. So we include the gas components also from the surrounding liquid, which is a small amount of air that is pre-dissolved in the aqueous phase under an atmospheric condition. Since air is mostly nitrogen and oxygen, we solve for one-dimensional radial diffusion of 3 gas components around a spherical bubble. We also consider the effects of linearly decreasing liquid pressure as we are following a bubble in a channel flow. And finally, we include the influence of surface tension as we are using surfactants in the experiments. Now let's look into the equations. We solve for radial diffusion of 3 gas components in the liquid phase to obtain the gas flux at the bubble interface. Such gas fluxes, as well as the liquid pressure and surface tension, have a coupled influence on the time change of radius of the bubble. The partial pressure of gas inside the bubble is connected with the boundary concentration of each gas component through the Henry's law. The equations are solved with Mathematica, and the calculated radius is plotted versus time for 8 millimolar SDS condition. We obtain very nice fitting of the data without any free parameters used for calculation. The partial pressure of CO2 and air inside the bubble clearly shows the gas exchange within approximately 30 milliseconds, perfectly predicting the 2 regimes of rapid dissolution and equilibrium. Air has slower dissolution rate compared to CO2, so after the CO2 bubble becomes an air bubble, its size does not change dramatically. The gas exchange idea can be further supported by comparing the calculations with experiments conducted using degas surfactant solutions. As you can see in the third plot, the equilibrium bubble size is much smaller if there is less pre-dissolved air in the liquid phase. Now that we confirmed the gas exchange idea as the main mechanism, we next try to explain the surfactant concentration dependence of the equilibrium radii. In the model, we have two additional parameters, the liquid pressure and surface tension. However, it appears from the graphs that changes in both the liquid flow rate and the surface tension do not influence the equilibrium size. As the model parameters cannot directly explain the concentration dependence, we need to come back to the experiments to double-check the details of how the experiments are set up in the beginning. If we look at the magnified view of the flow focusing region, we note that there is pressure difference applied at two sides of the gas stream that cuts the CO2 stream into a bubble. This means that at the bubble generation stage, there is already strong gas exchange as well as the surfactant coverage happening at the bubble interface. We thought that this could change the initial composition of the bubble depending on how many surfactant molecules are present at the interface. So, at higher SDS concentration, a thicker diffusion barrier is present and the generated bubble has less air inside. As you can see in this graph, if the bubble starts as a pure CO2 bubble, its equilibrium radius is smaller. Using this idea, we were able to predict the concentration dependence of the equilibrium radii as you can see from the fitted graph. To summarize this work, we started by observing non-disappearing CO2 bubbles in a microfluidic channel, and we were able to explain the main results using the multi-component gas exchange idea. We also figured out that surfactant molecules set up a diffusion barrier even at the bubble generation stage. Such a diffusion barrier controls the initial composition of the bubbles, which determines the final size of the bubbles. I would like to close this video with a final remark that our findings from this paper is later used to study the diffusivophoretic movement of bacteria under a CO2 gradient, and also inspired the hypothesis of gas-liquids-driven diffusiophoresis in a channel flow. Thank you for watching, and please let me know if you have any questions about this study.